These are the bone fragments. We receive it from the hospital, and it's basically it's a small piece of bone that I receive, and then I put it through a bone mill to obtain even smaller fragments, and then I just put those fragments in a flask and wait. My research particularly actually looks at cell source, which I can use so that we can immediately combine a scaffold material with cells and then immediately implant it into a defect, so that within a few hours you have a construct that could be used for bone regeneration. So actually it's about speeding up the conventional tissue engineering approach. Before you actually think about an in vivo experiment, you first make sure that, for example, cells do not die when they come in contact with a new kind of material. It needs to be biocompatible and not kill your cells. I work with human cells, so I would like to use an animal model that will not reject the human cells. So I need to go for immunocompromised nude animals, and there are mouse and rat animal models for that. And then you can either test whether your construct induces bone by implanting it, for example, in a location in the animal where usually there is no bone. Not all research that is done here will lead ultimately to an animal study and then further to an eventual product. So it's very complex, I'd say, if you combine materials, cells, and then maybe even growth factors, because you need to fine-tune and find the optimal combination, and that takes a lot of time and effort.